Recording full screen. So last time we talked about all the various different ways that we can affect regioselective and chemoselective control over a reaction. And we also talked a little bit about geometric selectivity in cis and trans double bond formation. So now we're going to talk about and I hope you will agree that what we have right here certainly counts as a complex molecule. Methyl homosecodaphniphyllate. Try saying that fast when you've had a few to drink. So this was isolated but from the bark of the Daphniphyllum genus, and the structure is known from X-ray. And it is a complex molecule containing 5 fused rings. And I would not like to count quite how many, but 1, 2, 3, 4, and then there's another kind of ring where it goes down here, where we would consider 5. Okay, so it is something. And this molecule was synthesized originally by Clayton Heathcock, and it is truly a beautiful bit of work. But before we actually get to that, I just want to talk about the proposed biosynthesis of this. I mean, I'm showing down here a whole bunch of other alkaloids, of which 6 is the one that we're addressing right now. But you can see that there's a lot of similarities in the family. In order to go from 6 to 5, you oxidize here. Well, we don't, the plant does, and then it cyclizes around onto the ester to give the lactone. And there are a whole bunch of other kind of transformations. In this case, it's got a much more complicated side chain, but again, the core is the same. 2 involves a rearrangement and a fragmentation of this bond here. Fragments to give this large 1, 2, 3, 4, 5, 6, 7-membered ring there. So there's quite some diversity within this system, but they all come from the same central thing. And in actual fact, they all start originally from this molecule, which I've actually not written down, but I'm pretty sure this is squalene. And of course, for all that we try and do selective reactions, plants and enzymes are just so much better at doing it than we ever will be, which is why a lot of enzymatic processes are just, stealing what plants do. So, oh good, I'm right, it is squalene. Just check that. Okay, so we do, in this case, a selective oxidation to put in these 2 aldehyde groups. And the proposed biosynthesis has initially this imine formation. So this could be either an amine from an amino acid, or very likely as well, it's an amine from something like pyridoxamine, which is a cofactor in a lot of biological processes, a lot of enzymatic processes. Anyway, the imine that's formed once it's bound to the enzyme is then reduced by NADPH, and the H is a reducing H, so it transfers in a conjugate addition sense to the imine. Now, the reason that the imine is reduced selectively over the aldehyde is because the enzyme makes it so, but it's also quite probable that the imine is protonated, which makes it actually more reactive. So you go from the conjugated imine to an enamine. And enamine is so called because you've got the ene part, which is the alkene, followed by the amine part, which is unsurprisingly the nitrogen. And then that NH lone pair cyclizes onto the aldehyde. Oh, no, sorry, my apologies. It does cyclize, but it cyclizes somewhere different. It actually, and this is why I have my notes, it actually does a conjugate addition. Okay, so you actually form a carbon ring. So this is the bond that is being formed in that step right there. And after that, that's when the nitrogen closes onto the aldehyde. Quite flustered today, my apologies. So the proposal was then, having got to this, oh, what happened there? Oh, my apologies. Having got through to this bicyclic system there, it actually undergoes a Diels-Alder reaction. This atom and this atom line up. Oh, sorry, nope, this atom and this atom line up. And, oh, what's going on today? And this atom and this atom line up and do the Diels-Alder reaction to get through to our tetracycle here. That then undergoes what's referred to as a Prinz reaction. To cyclize, and it loses the H. Okay? Now, there's something that's quite weird about this proposal. Something so weird that I think it's wrong. And that is this exoalkene here. And the reason that I'm saying that's weird is because not a single one of the natural products has an alkene here. Not one of them. And the standard way that biology does reduction of double bonds is this up here. You actually do a conjugate reduction. Okay, there is no palladium. There's no palladium and hydrogen in nature. Okay, and we will talk about an alternative in a minute. Okay, so I think this is almost right up to this intermediate. That intermediate I think is wrong. But that's what exists in the literature. Okay, so let's get to how Heathcock made this. So first, generated the lithium enolate using LDA. Okay, and then you do a conjugate addition, much like happened in nature, only onto a preformed 5-membered ring. And just want to point out that this conjugate addition, it's regioselective. Okay, we've talked about regioselectivity, and the way that they made this conjugate addition regioselective, or particularly regioselective, is heat. Higher T favors conjugate. What's really cool, though, is that by doing that, they generate an enolate, and that enolate is itself semi-stable. And therefore, they've generated a desired enolate at the expense of the amide enolate. So remember that an amide is less reactive than an ester. Therefore, amide enolate more reactive than ester enolate. Okay, so because the amide wants to reform quite a lot. So the amide attacks in the conjugate sense to the alpha beta unsaturated ester, and the ester enolate is then generated. And this is a favorable process overall because you've gone from the amide enolate to the esterinylate. The esterinylate can then be used to do an SN2 type alkylation, which goes on carbon because an alkyl bromide is a soft electrophile, prefers C alkylation. When we were talking about the extended enolate problem, we said that there's only very few reagents that are reactive enough and hard enough to actually react on oxygen. Okay, so we do our enolate alkylation and we get through to this system here. Now, this happens in a stereoselective sense, and we haven't talked about why it is stereoselective for the form that is caused here. We can do that as we progress on in the course, however. Okay, for now, it's not relevant. Okay, we then use dibal, and that is a chemoselective reduction of the ester, more reactive functional group, over the amide. Okay, so we reduce the ester group down to the alcohol, and then using strong base, this gets deprotonated and it substitutes the amide. Oh, no, that's not true. Sorry. I'm clearly losing my mind today. KOH does the hydrolysis to the acid, and then it lactonizes. Clearly having one of my bad days. Okay. So with that said, we get through to this lactone, and that lactone can then be reduced again. Okay, this time we use lithium aluminium hydride. Remember, we couldn't use lithium aluminium hydride earlier. Because it would reduce amide to amine. Okay, and that, of course, would be no good. So we needed to get through to this diol because we need to get through to this dialdehyde. Okay, and then we do the Swern oxidation to selectively oxidize to the dialdehyde. Remember when we talked about the Swern, we said it was a really clever way because the oxidation doesn't actually happen until you add the triethylamine. It's an excellent way to actually make a dialdehyde. Okay, if we were to use something like chromium trioxide, we would get back to the lactone. Okay, now this was just drawn wrong. This is the other aldehyde reacts first, but in the grand scheme of things, it doesn't really matter. But upon addition of ammonia, we get imine formation, we get enamine formation, and we get loss of water to get through to this system here, which can be considered an azodiene or a heterodiene, if we really want to call it that. Okay, and that heterodiene in the presence of acetic acid gets protonated. And now we get a really sweet reaction pathway because the protonation makes it more reactive and it undergoes a regioselective and chemoselective intramolecular Diels-Alder. Okay, it selectively reacts at the position we want. We have another alkene here, but it would give the wrong ring size and it would react a lot slower. So we actually have the selectivity for the alkene that we want. Okay, so we get our Diels-Alder reaction to make this tetracycle, and then we get the Prinz reaction, like I said, to give firstly a tertiary carbocation, and that tertiary carbocation loses a proton. This is really something, because if you look back to the previous slide, we started with here with a single ring. By adding ammonia followed by acetic acid in the same reaction pot, we end up getting to a compound with 5 rings. So we've had 4 new rings in one step. And that is pretty magical. How do we get through then to the final natural product? Well, first of all, hydrogen on palladium reduces the alkene, but it also removes the benzyl protecting group. Okay, I hope everyone knows this, but benzyl equals the benzene ring with a CH2, and it is removed by reduction. So that gets us through to the daphniphyllum core. Now we can use chromium trioxide to oxidize the primary alcohol all the way to the carboxylic acid. Remember, we started out with our chemoselective oxidations saying that it's hard to stop at the aldehyde, but it is actually very useful sometimes to make that desired carboxylic acid. And then finally, a Fischer esterification reacts on the carboxylic acid to give us the methyl ester. 9 steps, very elegant, very efficient. It's really clever. However, there is a slight issue, like I said at the very start. In the biosynthesis, they proposed this intermediate. This intermediate would then get oxidized across this double bond and then turned into the ester to make the methyl homosecodaphniphyllate. And this is the key intermediate of the biosynthesis that they called in one of Heathcock's papers protodaphniphyllene. And this is an intermediate that does not exist. It is a proposed biosynthetic intermediate that has never been isolated. But the alkane version, so that has been found. Okay, the alkene, easy to reduce chemically, very hard to do biochemically. So, how do we explain this? Well, this is something that I find even cooler. Okay? And what's hilarious about this, at least hilarious to me, is that this came about because of a mistake. Okay? It's very rare that science works this well for you, but it was actually done because they ran out of ammonia in the Heathcock lab. Okay, they were trying to do their biosynthetic. They were trying to prove their biosynthetic proposal, and so they had made this molecule that I have taken from a paper that's labeled 30. Okay, and they were trying to do the cyclization that I showed you on that initial biosynthesis step using ammonia. And they did actually successfully do that, but the yield was really low. And while they were trying to optimize this reaction, they ran out of ammonia. And so they went down the corridor and borrowed a bottle of ammonia in methanol from one of their colleagues. The bottle that they were loaned turned out not to be ammonia and methanol. It was actually methylamine. And that gave them the desired product, almost, in a much higher yield. When they used ammonia, I think the yield was something like 11%. But using methylamine, the yield was 65%, but no alkene. So, how can we do this? Well, first of all, let's just emphasize the fact that in the previous step I showed you, and the yield, I promise you, is not as good as you might hope it would be. Here, you've got a 65% yield, and you're forming 5 rings in one step from a completely linear precursor, which is amazing, in my opinion. So, how does this work, and why is there no alkene? Well, the mechanism follows exactly the mechanism shown on the previous page. Okay, this reacted with the ammonia, and the ammonia closes onto the unsaturated aldehyde. It is chemoselective for unsaturated aldehyde. The conjugation reduces the electrophilicity. So the imine forms onto the aldehyde, and then it forms the enamine under the conditions of the reaction. The enamine does the conjugate addition we proposed originally, and then you form the azodiene, which undergoes the Diels-Alder reaction, and it undergoes the Prinz reaction. Okay, so after switch colors, sorry. And I'm going to draw what happens after the Prinz reaction. Okay, so oh, what's sorry. Okay, so after the Prinz reaction, where you generate that tertiary carbocation, you have this compound. Oh, no, you very nearly have this compound. You have this compound. Sorry, I drew it to the wrong atom. And what's really interesting is that when you have methylamine, I'm just going to draw those hydrogens in, you have, oh, for goodness sake. You have 1, 2, 3, 4, 5, 6 atoms away from that carbocation. You have a hydrogen. And what ends up happening is that the lone pair on the nitrogen delivers the hydrogen to the carbocation. That then gives an iminium, which gets hydrolyzed by water to give the product 38. Now, most plant cells do not have ammonia just swimming around in there, but they've got a lot of amino acids. Okay, they're amino acids, so they have an alkyl group on the nitrogen. So it is far more likely, especially if it's something like lysine, perhaps, that what actually happens is that you generate the aminium, which then gets hydrolyzed to give the desired product. So protodaphniphyllate is actually not an intermediate that really exists. The real intermediate is compound 38. Okay, so that is the end of part 1 of this course on control and selectivity for total synthesis. So I am going to, yes, keep those.